All right. Well, next up, we have Prashant, who is from the College of Agricultural, Food, and Environment in the Department of Veterinary Sciences. So take it away.
Good afternoon to all. I'm Prashant Purushottam Damke from Department of Veterinary Science, and I would like to thank organizers for giving me this opportunity to present my work. So today I'm going to talk about how a gastric pathogen which lives inside our stomach is able to translocate its DNA directly into the human cells. And the title of my talk is like horizontal DNA transfer from Helicobacter pylori to humans. So the horizontal transfer of DNA from microbes to eukaryotes has been shaping the evolution of eukaryotic and prokaryotic genome from millennia. And there are several mechanisms of this DNA transfer present in the nature. So, for example, like ingested prokaryotes and eukaryotes and various endosymbionts like mitochondria and chloroplast are able to translocate their DNA directly into the human or eukaryotic cell nucleus. Also, many viruses and certain bacteria are able to translocate their DNA into the eukaryotic cell nucleus, which can then get integrated into the chromosomal DNA of the eukaryotes. And this process has been shaping the evolution of the genomes since millennia and has been directly responsible for genome instability and cancer progression and induction of immune responses in eukaryotic host cells. More importantly, this DNA transfer system are a very rich source of various genetic engineering tools and have been extensively used for plant and human and eukaryotic genome modification. Also, they have been extensively used to deliver specific genes and vaccines to various eukaryotes. So out of this, I'm particularly interested in studying how certain bacteria can translocate their DNA into the eukaryotic host cells. This process is quite rare in nature, and there are not many examples of such process occurring. For example, only few bacteria are known to translocate their DNA into the eukaryotic host cells. For example, Agrobacterium tumefaciens and Helicobacter pylori are known to translocate their DNA. So out of these, the best studied system is that of Agrobacterium tumefaciens. So Agrobacterium tumefaciens is a plant soil bacterium that causes crown gall disease in the plants. So this Agrobacterium tumefaciens has a TI plasmid, and the virulence region of this TI plasmid encodes for several DNA processing and secretion system genes. So upon induction by the plant phenolic compounds, the secretion system proteins of this TI plasmid assemble a D4 secretion channel at the bacteria and plant cell interface. Then the DNA processing proteins of this TI plasmid, particularly the BLD2 protein, excise tDNA region from this plasmid at the left and right border, and then that whole nucleoprotein complex is regulated at the secretion channel, which is then subsequently translocated into the plant cell, which then goes to the plant cell nucleus, where the DNA from the TI plasmid can get integrated into plant cell chromosome by illegitimate recombination. So a similar kind of secretion channel or a secretion system is also found in Helicobacter pylori, which is the model system which I work on. So this Helicobacter pylori bacteria, it lives inside our stomach, and upon contact with the gastric epithelial cells, it assembles a secretion channel similar to the D4 system of Agrobacterium tumefaciens. And then this secretion system is known to translocate various effector molecules directly into the human cells, including protein molecules, lipid molecules, and peptidogram molecules. So recent evidences have indicated that the Helicobacter pylori is also able to secrete DNA directly into the human cells. So monitor this secretion of DNA into the human cells. We developed some assays where we look for the induction of DNA sensing pathways in the human cells. So we specifically look for induction of TLR9 and cGAS-STING pathways in human cells. So TLR9 are the endosome bound receptor which can detect the presence of microbial DNA in the human cells. And the cGAS is a sensor which can detect presence of aberrantly present DNA in the cytoplasm of human cells. So we see that both these pathways are getting activated by Helicobacter pylori in CAG type 4 secretion system dependent manner. And using similar assays, we found out that the DNA sensing, the DNA translocation is directly dependent upon the interaction between the bacteria and the human cell. So next, to provide a direct evidence that Helicobacter pylori DNA is getting translocated into the human cells, we purified the C-gas protein complexes, and we purified the DNA that was co-precipitated with the C-gas, and we did the sequencing. And when we looked at the enrichment of the peaks specific to Helicobacter chromosome, pylori chromosome, we observed that the enriched peaks were specifically located around the ORIC region of the Helicobacter pylori chromosome. Then in the same line, we observed that upon addition of DNA replication inhibitor, we see loss of TLR9 activation, indicating that the DNA translocation is dependent upon DNA replication. Using reverse genetic approach, we identified several Helicobacter pylori proteins that are directly involved in chromosomal replication and declination, that are directly involved in DNA transfer from Helicobacter pylori to human cells. Particularly this FTSK protein, which recognizes this defect sequence, is very important for the DNA transfer system. Again, using the same reverse genetic approach, we identified a DNA relaxase that can cleave DNA at specific certain sites, that is also directly involved in the DNA transfer from Helicobacter pylori to humans. And this is some biochemical analysis to show that this DNA relaxase actually cuts DNA at particular sites. So after also we show that using protein-protein interaction studies, we show that this FTSK protein molecule is directly responsible for recruiting the excised DNA directly into the CAG secretion channel. So after having a basic idea about the possible mechanism of DNA translocation by Helicobacter pylori, we were interested in looking, can we use this system to deliver some specific DNA molecules into the human cells? So to find out that, we designed, we integrated a reporter gene construct into Helicobacter pylori chromosome, which contains enhancer and promoter and polyase sequences optimized for eukaryotic gene expression. So when we infected this wild type and CAG negative strain, we were able to see the expression of the nanoluciferase gene upon a wild type strain infection. Also, when we replace this nanoluciferase reporter gene construct with another reporter molecule such as neon green and M scarlet, and when we infected the wild type strain to these human cells, we were able to see that this DNA can get translocated into the human cells and start expressing. So to get a more quantitative data on this, we did flow cytometer analysis, and you can see here that upon a wild type infection, there is a significantly higher number of fluorescently positive cells compared to the CAG negative strains. Next, certain Helicobacter pylori strains has plasmids. So we were interested if this system can also deliver plasmid DNA molecules into the human cells. But surprisingly, when we integrated the same reporter gene into the Helicobacter pylori plasmid, there was no transfer of this plasmid. When we looked at the sequences, there are no recognition sequence for the DNA relaxase or the FTSK protein in these plasmids. So I integrated those recognition sequences of those two key DNA processing proteins in these plasmids, various mutation and combinations. And then we were able to see that, okay, now this plasmid is able to deliver to the human cells, albeit with different efficiency. And we found out that the recognition sequence of the FTSK protein is most efficient at driving the transfer of DNA from Helicobacter to human cells. So, so to summarize, basically, we show a very novel pathway that Helicobacter pylori is able to deliver its DNA to the human cells, and we identified a basic protein machinery that is required for delivering, processing and delivering these DNA molecules. And most importantly, what we see that we can exploit this system to deliver a specific reporter gene molecules or DNA construct to the human cells. And most probably in future, we can use this system to deliver certain kind of, say, drug molecules or vaccine molecules or some RNA molecules directly into the human cells. And also there are a lot of open questions about the mechanism mostly, like we don't know what exactly is the DNA processing complex that is being assembled at the in Helicobacter pylori. We also don't know if some of these proteins or DNA, how they get translocated into the human cells. We also don't know what exactly happens to the DNA when it gets translocated into the human cells. So there are a lot of open questions, some of which I'm working currently in the lab. And so again, thanks for the organizers to give me this opportunity. And I would like to thank my lab at the Department of Veterinary Science. Thank you very much.